Hi, everybody. This is lecture 20. We're in the second half of chapter 13, How Populations Evolve, part 2. So if you remember from the first half of this chapter, we were talking about Darwin's theory of natural selection, a mechanism of evolution, which was the process of change over time. So now we're going to be getting a little more into that and looking at some other situations as well. So Darwin proposed natural selection as the mechanism of evolution. Remember that evolution is the process, natural selection is the mechanism by which evolution is happening. So Darwin's greatest contribution to biology was his explanation of how life evolves. Remember, we knew that evolution was happening. We knew that change over time was happening, but we didn't know how it was happening until Darwin, when he explained with his theory of natural selection. Although, again, remember, he didn't actually call it natural selection, but he described that process. So Darwin thought that species formed gradually over long periods of time. So he knew that he would not be able to study the evolution of new species by direct observation. Now, when we talk about speciation, we'll talk about how there are exceptions to that, but generally Darwin was correct. Typically, evolution happens over a very long period of time. We're talking thousands, hundreds of thousands, maybe even millions of years. And so usually you can't actually just sit there in one lifetime and directly observe observation. So Darwin knew that he was going to have to be kind of creative of how he was going to do this. Now, one of the ways that we can get insights into natural selection is by looking at artificial selection. So the idea of artificial selection is that this is a type of selection. It's similar to natural selection, but it's where humans have meddled with things, right? Where humans have gotten involved and they've modified species through selective breeding. This is something you're familiar with, even if you don't know that you are. If you know about domestic dogs, they're a great example of artificial selection or selective breeding. So domestic dogs are a genetic mess. They're a cute, adorable genetic mess, but domestic dogs are a genetic mess. Domestic dogs are all one species. So everything from your Doberman to your Pit Bull to your teacup Chihuahua, all the same species, just different breeds, all the same species. And this is because of artificial selection. So, for depending on, which scientific study you look at, domestic dogs evolved somewhere between 10,000 and like 100,000 years ago. From wild canines, possibly from wolves, and due to some, mutualistic relationship there, humans started artificially breeding dogs. And it could have been for, the ones that were the friendliest or the ones that bark the least. Or when we start getting into, the modern ways that we have selectively bred dogs, the ones that have the shortest noses or the fluffiest tails, or their face looks a certain way, or they stand a certain way, or they can trot around the arena in a certain way. And we selectively breed them. So we purposely pick two dogs. Like, I want one with a really squished up face because I don't want that dog to be able to breathe. And so my aunt's a national dog show judge, so I have some stories if you're interested. But they'll be like, that squishy face, that is what's going to look best on stage. So I'm going to take a male with the squishiest face and a female with the squishiest face. I'm going to breed them. And then you do it over and over again until you get just the type of squishy face that you want. And yes, domestic dogs are incredibly inbred because of this, but they have some real squishy faces that apparently look real good when you bring them to dog shows. And so that is one example of artificial selection that is fairly easy to watch quickly, because there is a whole line of work. I have some cousins who are dog breeders. That's what they do for a living, is they purposefully just breed dogs to get the traits the dog show audience wants. And so this can happen fairly quickly when we are selectively breeding generation after generation for a certain trait. So it's not natural selection because we are purposely picking these traits that we want, but it's the same concept is what could be happening. It's just happening a lot quicker than natural selection is. So we're able to observe artificial selection and draw some inferences about natural selection from it. Another good example of artificial selection is food, by the way. We have been artificially breeding food, because if you've ever looked at the wild types of some of the food you eat, they're crazy, right? So we selectively breed them to have less seeds, no pokey things are gonna poke us in the roof of the mouth when we eat them, resistance to pests, things like that. If you look at the pigeon on the screen, this is another example of artificial selection. We can breed pigeons to have cute little tails, little fluffy legs, frilly backs, things like that. So artificial selection is very common. We do it with a lot of different animals and plants, and it happens a lot quicker usually than natural selection. So we can make some inferences from it. So back to natural selection, it is important to emphasize 3 key points about evolution by natural selection. So one of them is that although natural selection occurs through interactions between individual organisms and the environment, individuals do not evolve. Think back to the Pokemon that I showed you in one of the previous lectures. Individuals themselves do not evolve. Remember that when we're talking about evolution, we are talking at the population level. So it's that group of organisms that evolves over time. And this makes sense when you think about the more specific definition of natural selection or of evolution, and then it's the change in the gene or allele frequency in a population over time. So individuals do not evolve, populations do. Also, it's important to keep in mind that remember that natural selection is an editing process. It's not a creative process. We're not really creating anything new. We're editing what already exists. And so we're either amplifying or diminishing heritable traits. Remember, the whole use it or lose it idea was not correct. In order for natural selection to be happening, we're talking about heritable traits. They have to be things that can actually be passed on to offspring. And also remember that evolution is not goal-directed. There is no perfect organism. And that makes sense when you think about why natural selection is happening, and specifically about adaptations. So the environment is constantly changing. We are in a period right now where we can actually watch that happening. When you look at climate change, we can watch the temperature going up year after year. We're watching, the effects of climate change in terms of the pandemic and increased weather events like hurricanes, sea level rise with the king tides. These are things that we can observe. And so the environment is constantly changing. And so there can't be a perfect organism, because the moment you get the perfect organism, the environment's going to change again. And that perfect organism is no longer perfect anymore. And so evolution is not goal directed. It's whoever, happens to have the best traits for the given environment at the time, keeping in mind that that could change in an instant. So scientists can actually observe natural selection in action. So we're going to take a look at that. So like I mentioned, these examples of evolutionary adaptation highlight 2 important points about natural selection. So natural selection, again, is more of an editing process than a creative mechanism. And natural selection is contingent on time and place. So it favors the heritable traits in a varying population that fit the current local environment. So we're not making anything new. We're just editing what already exists. And we're editing what already exists based on whatever is happening in our current local environment at the time, keeping in mind that that can change at a moment's notice, or maybe over thousands of years. It all depends. So let's look at the evolution of populations. So mutation and sexual reproduction produce genetic variation that makes evolution possible. Remember, one of Darwin's inferences, or one of his observations, was that populations vary genetically. There are different traits, right? Natural selection couldn't happen if every single organism was genetically identical. Natural selection happens because individuals are different. They have different genes that give them different adaptations, which then make certain individuals more likely to survive and reproduce than others. Natural selection hinges upon that genetic variation. That is also why when we're talking about conservation, it's really important that a population be genetically diverse. The more genetically diverse a population is, typically the healthier that population is, and the more likely they are to survive some kind of catastrophic event. Because if you think about, say, a pandemic coming in, and there are certain individuals whose genes make them more susceptible or less susceptible, right? Say you had individuals, like the entire population was almost genetically identical, and they were susceptible to the pandemic. That would wipe almost everybody out. This happens a lot in crops. We see that a lot in crops where things have been really interbred. And so a lot of the crops are genetically identical or very, very similar, and the plague will come in and wipe out the whole crop. If you have a genetically diverse population, it's much more likely that there's somebody or a small subset in there that are going to be able to survive. So genetic diversity is super, super important, especially when we're talking about conservation. So organisms typically show individual variation. You know, that's the idea that we're all genetically different from one another for the most part. And so not only are they going to look different, that their genetics are going to be different as well. Now, however, in The Origin of Species, Darwin could not explain the cause of this variation among individuals and how those variations were passed from parents to offspring. Remember that Darwin was operating before Gregor Mendel did his experiment with the peas. So it was Gregor Mendel who really explained that process of heredity. And so Darwin knew it was happening, but he didn't know how. So he was able to explain, like, oh, this is happening somehow. And then Gregor Mendel came in later and explained how that was happening. So individuals have unique genomes. Sometimes mutations occur that help to create these unique genomes. You know, there's chromosomal duplication and then also sexual reproduction. So sexual reproduction is really important to creating genetic diversity because we get crossing over. We get random fertilization where you don't know, you know, which sperm is going to you know, match with which egg, and then also independent assortment where we had the homologous chromosomes lining up and we didn't know if it was going to be mom on one side, dad on this side, or if it was going to be switched. So different causes of genetic diversity. On rare occasions, however, a mutated allele may actually improve the adaptation of an individual to its environment. So we quite often think of mutations as bad things, but yeah, like think of the Ninja Turtles. Like sometimes mutations can be pretty cool. Oh, teacher, that was a bad joke. But mutations can actually be beneficial. So, you know, we can have mutations that make a certain crop resistant to pests or more drought tolerant. Or say you have an organism that had a mutation that caused them to grow more fur, and then all of a sudden the environment turned really, really cold. Now they're going to be doing better. This kind of effect is more likely when the environment is changing, such that mutations that were once disadvantageous are now favorable. So we're usually not creating something new, but rather there was some kind of trait that we were only seeing in really low levels in a population, you know, something like being really, really furry if they were living in a warm environment. And then all of a sudden it turned cold. Now that adaptation that already existed, now it's favorable. So now we're going to start seeing it more. So that's usually what we see happening. So let's look at how evolution occurs within populations. So again, remember that a population is a group of individuals of the same species that live in the same area and interbreed. We can actually measure evolution as a change in the prevalence of certain heritable traits. So, you know, the genes or alleles in a population over the span of generations. Different populations of the same species may be geographically isolated from each other to such an extent that exchange of genetic material never occurs or very rarely occurs. And we quite often see this in lakes or islands, or more and more now we're starting to see that with the growth of cities. So cities are separating animals and plants, you know, the construction of roads, walls, things like that. So a lot of human edifices are also separating populations now. Another vocab word for you, the gene pool. The gene pool consists of all copies of every type of allele at every locus in all members of the population. So we're basically talking about all the genes that exist in a population. That's the gene pool. And then microevolution. So microevolution is a change in the relative frequencies of alleles in a population over a number of generations, and it's evolution occurring on its smallest scale. So the key word here is population. So microevolution is basically when we're talking about evolution on the scale of the population. So now we're going to jump into a little math equation. The Hardy-Weinberg equation can actually test whether a population is evolving. So to understand how microevolution works at the population level, we're going to look at a simple population in which evolution is not occurring, so the gene pool is not changing. So I'm going to give you an example here, and we're going to apply the Hardy-Weinberg principles and equation to this population to see whether or not evolution is happening. So consider an imaginary population of iguanas, and they differ in foot webbing. So the webbing in between their little toes. And let's assume that foot webbing is controlled by a single gene, and that the allele for non-webbed feet is completely dominant over the allele for webbed feet. So we'll represent non-webbed feet with a capital W, and we'll represent webbed feet with a lowercase w. So if you take a look, here is an individual with no webbing, and here is an individual with webbing. Yes, they are mating because we are going to examine their offspring now. So the shuffling of alleles that accompanies sexual reproduction, like we just saw those iguanas doing, does not actually alter the genetic makeup of the population as a whole. So no matter how many times alleles are segregated into different gametes and united in different combinations by fertilization, the frequency of each allele in the gene pool will remain constant unless other factors are operating. So things are going to stay the same unless something else is going on. So let's take a look at that. So this equilibrium, the fact that things are going to stay the same, the frequencies of alleles in the entire population is not going to change, is known as the Hardy-Weinberg principle. It's named after two scientists, Hardy and Weinberg, because we like naming things after ourselves, who derived it independently in 1908. So this is the Hardy-Weinberg principle. This is the equation here. So if you take a look, we have the frequency of the homozygous dominant genotype, which is over here represented by p squared. We have frequency of the heterozygous genotype, so we've got 2pq, and the frequency of the homozygous recessive genotype, so that is q squared. When you put that together, that equals 1. Think of this as in a percentage, so 1 is 100%. So if you look at P is going to represent the homozygous dominant allele. So here's P again, and then Q is going to represent the homozygous recessive allele. That's going to make more sense, the difference between P's and P squareds and things when we do the example in just a minute. So to test the Hardy-Weinberg principle, let's look at 2 generations of our imaginary iguana population. So I'm going to show you figure 13.10b in just a minute, but it's going to show the frequencies of alleles in the gene pool of the original population. And then from those genotype frequencies, we can calculate the frequency of each allele in the population. So remember that our genotype frequencies are going to include 2 different alleles, and so there's different combinations of those alleles. So let's take a look at that. So we have our phenotypes here. So we have our homozygous dominant individuals that are big W, big W. These would be non-webbed. We have our heterozygous individuals that are big W, little w. They would also be non-webbed. And then we have our homozygous recessive individuals who are little w, and they have the webbed feet. Now in our example, there's a population of 500 animals. So we have 500 iguanas, 320 of them are homozygous dominant, 160 of them are heterozygous, and 20 of them are homozygous recessive. So let's take a look at the genotype frequencies. So again, genotype frequencies are referring to homozygous dominant, heterozygous, or homozygous recessive. So taking into account both the alleles in order to get the genotype. So we're going to take a look at our genotype frequencies here first. So remember, they're out of 500 individuals. So each of these fractions will be over 500. To get the genotype frequency, you're simply going to take the number of individuals with that genotype and divide it by the total number of individuals in the population. So if we are looking at homozygous dominant, there are 320 homozygous dominant individuals divided by 500 total individuals in that population. That's going to give us a genotype frequency of 0.64. You can also think of that as 64% that are homozygous dominant. Let's take a look at our heterozygous individuals. So there are 160 individuals in this population that are heterozygous. So we're going to divide that by 500 total individuals. That gives us a genotype frequency of 0.32. And then if we look at our homozygous recessive individuals, there are 20 of them. So we're going to divide 20 homozygous recessive individuals over 500 total individuals in that population, giving us a genotype frequency of 0.04 for our homozygous recessive genotype. So now we need to calculate the allele frequencies. So if you remember, we had the idea of p squared and p. So p is going to represent, in that Hardy-Weinberg equation, p is going to represent the dominant allele. So in this case, that is uppercase W. Q is going to represent the recessive allele. So in this case, that is lowercase w. So in order to get the allele frequencies, we've got to do a little more math. So we want to know how many big W's and how many little w's are present in this population. So remember, if we have 500 total individuals, that means we're looking at 1,000 alleles, because each individual is going to have two, for the most part, should have two different alleles, right? Or two alleles. So they should have, either big W, they get a big W, or they get a little w, but each one is going to have two alleles. So if we take a look, there are 320 of them that have big W, That means the homozygous dominant individuals are contributing 640 big W's, right? There's 320 that have big W. So there's 320 here, there's 320 here, right? So if you do that math, 640 from our homozygous dominant individuals. If we look at our heterozygous individuals, there's 160 of them that each have a big W and each have a little w. So our heterozygous individuals are contributing 160 big W's and 160 little w's. Then if we take a look at our homozygous recessive individuals, there are 20 of them, each contributing 2 little w's to the gene pool. So we get 40 little w's from the homozygous recessive individuals. So if you take a look here, 640 big W's from our homozygous dominant individuals and 160 big W's from our heterozygous individuals. That gives us 800 big W's in this population. If you take a look over here at the little w's, the recessive allele, there's 160 little w's coming from our heterozygous individuals. There's 40 little w's coming from our homozygous recessive individuals, for a total of 200 little w's in this gene pool. So to get the frequencies, we're again going to divide by the total number of alleles in that gene pool. That is 1,000. Remember, there's 500 individual animals, and there's 1,000 alleles, since each individual has two, one from mom and one from dad. So we have 800 big W's divided by 1,000 total alleles. That gives us an allele frequency of 0.8 for big W. For little w, our recessive allele, there's 200 of them, right? 160 from the heterozygotes, 40 from homozygous recessive individuals. So 200 divided by 1,000 is 0.2 as our allele frequency for little w. So again, remember in this example, big W represented P. P is the allele frequency of the dominant allele. Q is the allele frequency of the recessive allele. So 0.8 for big W equates to P as 0.8, and Q is representing our little w, so Q equals 0.2. So you could go back into that P squared plus 2PQ plus Q squared equation, and you could calculate out what that comes to. So gametes reflect allele frequencies of the parental gene pool. So we're going to take this a step further now. And so we know that the allele frequency of W, represented by P in our Hardy-Weinberg equation, is 0.8. Little w's are 0.2. So if we are looking at heterozygotes here, right, big W, little w, we can actually do a cross here to see what the frequencies of the offspring would be. And so you'll notice we're starting to get into the equation here. So here's P squared, right, where we have one parent with P, right, big W. So big W is actually P squared in that equation. And that gives us P squared equals 0.64. If you get a little w from one, big W from the other, that is PQ, 0.16. Same thing down here. And then if you get w, little w, that is q squared equals 0.04. So this is where our equation is coming from, right? If you're getting a big W and a big W from both your parents, big W, that equals p squared because you're timesing them. This is the 2pq part here because there'll be two that have a p and a q. And then this is where q squared comes from because there'll be one square here that has little w. So we've got our genotype frequencies, and we've got our allele frequencies in the next generation. Notice they're the same, right? So our frequencies have not changed when we go to the next generation. So if a population is in Hardy-Weinberg equilibrium, allele and genotype frequencies will remain constant. So here, when we did that cross between the heterozygous individuals, we ended up with the next generation having the same frequencies, the same genotype frequencies, the same allele frequencies. And if you kept doing this over and over again, if the population is in Hardy-Weinberg equilibrium, you're going to keep getting the same results every single time. So that's what we're seeing here is with this population of iguanas, we're getting the same genotype frequency and the same allele frequency generation after generation. So that's telling us that this population of iguanas is in Hardy-Weinberg equilibrium. But I want you to think about real life and how often populations are actually going to be in Hardy-Weinberg equilibrium. It's not often. So that tells us that there's something other than the reshuffling processes of sexual reproduction that are required to change allele frequencies in a population. Because think back to the example we just did, these alleles are shuffling, right? We still have sexual reproduction. We still, we're getting crossing over, independent assortment. We're getting, where we don't know which sperm is going to match with which egg. And our frequencies are still staying the same. So if sexual reproduction is not doing it, there has to be something else at play. So for a population to be in Hardy-Weinberg equilibrium, it must satisfy 5 main conditions. So it has to be a really, really big population. There has to be no gene flow between different populations, no mutations, no random mating, yes, random mating, yes, random mating, and no natural selection. So very large population, no gene flow, no mutations, yes, random mating, no natural selection. And I want you to think about how often all 5 of these are met. It's not often. So there are not that many populations actually in Hardy-Weinberg equilibrium. So what is going on? Rarely are all 5 conditions met. So allele and genotype frequencies are often changing, but the Hardy-Weinberg equation can be used to test whether evolution is occurring in a population. So Hardy-Weinberg equilibrium is rarely ever happening. So what are these other things going on, right? What is causing evolution? So let's take a look at mechanisms of microevolution. So we know about natural selection, right? We learned about natural selection in the last lecture. So we're also going to look at the concepts of genetic drift and gene flow that can cause microevolution. So if the 5 conditions for the Hardy-Weinberg equilibrium are not met in a population, the allele frequencies may change. Mutations are something that would probably first come to mind to you since we've studied those before, but mutations are actually fairly rare and they're pretty random, and they actually usually have little effect on the gene pool. And non-random mating, so when individuals are being choosy, may change genotype frequencies, but usually has little impact on allele frequencies. So therefore, the 3 main causes of evolutionary change are natural selection, genetic drift, and gene flow. So we've talked about natural selection already. So let's talk about genetic drift and gene flow. So in genetic drift, chance events can cause allele frequencies to fluctuate unpredictably from one generation to the next. And the smaller the population, the more impact genetic drift is likely to have. If you think back to one of the requirements for Hardy-Weinberg equilibrium, it was that they have a very large population. That's because large populations are less susceptible to things like genetic drift. There's just so many of them that you can change, a couple individuals and it's not going to make that much of a difference. If you have a really small population though, and even 1 or 2 individuals something happens to, that's going to make a big difference. Think about, I always tell people to think about sample size when you're looking at any kind of statistic, because, if I'm talking about, 2 individuals are going to get this disease, right? If we're talking about 2 individuals out of a population of 7 billion, 2 individuals isn't really going to change that population of 7 billion. But if we're looking at a population of 10 individuals, 2 individuals getting this illness out of 10, that's a big deal. So genetic drift affects small populations much more. Genetic drift is caused by things like catastrophes, such as hurricanes, floods, fires that may kill large numbers of individuals. So it leaves a small surviving population that's unlikely to have the same genetic makeup as the original population. Again, especially true for small populations. And the bottleneck effect leads to a loss of genetic diversity when a population is greatly reduced. So let's take a look. This is the idea of the bottleneck effect. So take a look at the original population. We have purples and greens that are, fairly equal to each other, and then we have a few oranges in here. And then there's going to be some kind of event that's going to kill off a lot of the population. So only a few of them are going to survive. The ones that survive are the ones that make it out of the bottle. And say they're being shaken into this cup. So the ones in the cup are the ones that survived. So if you take a look at all of these individuals, each of our marbles is representing an individual. The colors are representing the different genes that they have. Only 5 of them survive. Out of all of these, only 5 of them survive. And this can be, fairly random depending on the catastrophe. And they're going to go through this bottleneck effect, right? So only 5 of them survive. And now take a look. Out of that surviving population, we have 4 purples and 1 green. So this does not represent the original population at all. That's the idea of the bottleneck effect. So genetic drift is also likely when a few individuals colonize an island or other new habitat. This produces what's called the founder effect, since they're the founding members of this new population. Again, the smaller the group, the less likely the genetic makeup of the colonists will represent the gene pool of the original population that they just left. And the founder effect explains the relatively high frequencies of certain inherited disorders among some populations, in some human populations, established by a small number of colonists. Because if there is some kind of, inherited disorder, if you're talking again about a large population, that inherited disorder is not going to have that much of an effect. But think about if they move to a new location, like an island. We have a group that colonizes an island. So now instead of having thousands of individuals, we have dozens of individuals, and 1 or 2 of them have this inherited disorder. Now they represent a much larger percentage of the gene pool. And so that inherited disorder is much more likely to have an effect on the overall population in this new, smaller population than it was in the old, larger population. So in gene flow, the allele frequencies in a population will also be changing. And gene flow is where a population may gain or lose alleles when fertile individuals move into or out of a population, or when gametes, like plant pollen, are transferred between populations. Remember, with plants, some of that pollen can travel by air or on the backs of individuals or in the poops of individuals. And so plant pollen can actually get dispersed quite a bit more. Instead of with animals where the individual has to move. And gene flow tends to reduce differences between populations. So the idea behind gene flow is that we have 2 different populations, and we could get an individual or a small group of individuals from population A that decide to leave and move to population B. So especially if they've been separated for a long amount of time, this could introduce genes that population B didn't have. So the genetic diversity of population A could actually go down, and the genetic diversity of population B could actually go up, depending on which genes those individuals are carrying. But that's the idea of gene flow, is that some individuals leave group A and then they go to group B, and they're going to breed with the individuals in group B, introducing new genes. So that's the idea of gene flow. So natural selection, though, is the only mechanism that consistently leads to what we call adaptive evolution. So genetic drift, gene flow, even mutations could each result in microevolution, but only by chance could these events improve a population's fit to its environment. In natural selection, though, only the genetic variation produced by mutation and sexual reproduction results from random events. So the process of natural selection, in which some individuals are more likely than others to survive and reproduce, is not random. Because of this sorting, only natural selection consistently leads to adaptive evolution. So when we were talking about genetic drift and gene flow, that's happening mainly as the result of random events. But remember that natural selection is not random. So natural selection is, I mean, you just so happen to have the adaptations for the environment, but it's not random who is surviving and who is reproducing. It's whoever happens to have those adaptations, but whoever does have those adaptations that fit the current local environment the best, those are going to be the ones surviving and reproducing. So natural selection, it's not random who is surviving and reproducing. So if you take a look at this bird, this is the blue-footed booby. It's got these bright blue feet, which when you first look at it, you're like, wow, those are some bright blue feet. That's going to attract some attention. That could be good. That could be bad. It could be bad as in like, those might look delicious. And so some predators might want to eat it. But at the same time, they could also be real attractive to some mates. And so we see this a lot when we talk about genetic dimorphism, where the males and the females look different. It's typically the males that are bright and flashy. Think of peacocks, think of lions with their big manes. Birds are a great example because they have some adaptations to attract those girlfriends. It's usually the males trying to attract the females. It's because, one male could mate with multiple females, but the female can, usually only be pregnant one at a time. And so males have these real flashy adaptations that help to attract some mates. And so whichever one of these blue-footed boobies has the brightest, flashiest feet is going to be the one that reproduces the most. And that's not random, right? The females are actually choosing who has the nicest feet for their mates. So natural selection can alter variation in a population in 3 different ways. The first is stabilizing selection, which favors the intermediate phenotype. So if you look at the blue line here, this is the original population. So notice there's a much larger spread here. We have our kind of outlier phenotypes on either side. Again, this is representing the phenotypes that you see in a certain population. So much more laid out spread in the original population. In stabilizing selection, it's going to select the phenotypes in the middle. So after natural selection, we're going to see fewer phenotypes, and it's going to favor the ones in the middle. So an example of this is robins. Robins typically lay 4 eggs. Larger clutches may result in malnourished chicks. There's just too many of them to feed. And smaller clutches may result in no viable offspring, right? If you only have 1 or 2 eggs, they might just not survive. And so 4 eggs seems to be the sweet spot right there in the middle. So that's an example of stabilizing selection. We're choosing the intermediate phenotype, that one in the middle. Another example is directional selection, which shifts the overall genetic makeup of the population. It actually acts against individuals at one of the phenotypic extremes. So if you take a look, the dotted line here is the original population. And then after selection, there's one extreme trait that's favored. It's whatever one's on the right here. The entire thing is going to shift. So it's going to shift the entire genetic makeup of the population over to favor whatever trait this is over here on the right-hand side. So moving the whole thing. And then disruptive selection typically occurs when environmental conditions vary in a way that favors individuals at both ends of the phenotypic range. So again, the dotted line here is the original population. The solid line is the population after selection. You'll notice that we had that original bell curve here, but then after selection, whatever traits are here on the extreme ends are what are being favored in disruptive selection. You can think of, this is being disrupted in the middle because favorable, 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 ooh, not favorable, and then we're favorable again on the other side. So that's disruptive selection. Sexual selection may lead to phenotypic differences between males and females. We briefly touched on that just a minute ago when I introduced you to the idea of genetic or sexual dimorphism. Sexual dimorphism is where males and females look different from one another. Sexual selection is a form of natural selection in which individuals with certain characteristics are more likely than other individuals to obtain mates. Think about peacocks. right? If you look at the male peacock, they have those bright, brilliant tails, whereas the peahens are actually kind of bland and boring. You see this a lot in birds. A lot of male birds are really colorful, have really big feathers, where the females are kind of small and drab. The reason for that is sexual selection. If you think about a peacock's tail, it is big, it is flashy, it is hard to live with, it gets in their way, it drags on the ground. The zoo guests all go and pluck the feathers out of their bums. And they're hard to live with. So why is it worth it? Because if you have that big shiny tail that gets in your way and makes it harder to live and you're still alive, that's telling the females that you have some nice genes. So when we see those big flashy adaptations, you know, especially in male birds, we see this in other species as well. Fish, you'll see this a lot in, even things like a lion's mane. You know, if it is that big, it can be hot, it can be heavy, it can get in the way. If that male is still alive, that tells the female they've got some pretty good genes. So that's who the females want to mate with. So the females will actually choose the peacock with the biggest, brightest feathers, the male lion with the biggest, heaviest mane. You know, the blue-footed booby with the bluest, brightest feet. That's the idea of sexual selection. That adaptation is telling the females, if that male is still alive, he's going to make some great babies. So in many animal species, males and females have secondary sexual characteristics. That is secondary sexual characteristics are things like that big bright tail, the manes on the lions. Again, this is called sexual dimorphism, where males and females look different. Ooh, although we have started seeing some female lions with manes, right? It's interesting. We're not exactly sure why. It's a new thing. You can read up on it. In some species, individuals compete directly with members of the same sex for mates. So you see this a lot in hoof stock, you know, things like deer, where you'll see two males going at it, trying to compete over a female. This is called intrasexual selection, intra with an A here. So within the same sex, that is usually seen in male-male competition. So we rarely see females fighting over males. It happens sometimes, but not in very many species. It's more common for two males to be fighting. So male-male competition is an example of intrasexual selection. That is just another way to show the females, you know, I fought, I won, that means I have the best genes, you should mate with me. Contests quite often involve physical combat, but quite often they're even things like ritualized displays, so they don't always have to hurt each other. A lot of them do. A lot of these rituals involve males getting hurt, but they don't always. Sometimes it can just be some kind of dance or some kind of other display like that. In a more common type of sexual selection, there's intersexual selection. So look at the E here, intersexual selection, and that is between the sexes. So this is also known as mate choice. That is where, you know, typically the females are selecting their mate. An example of this is bowerbirds. So if you take a look in the background here, this is a type of bird found in Australia. Well, this is not the bird found in Australia. This is the bower of a bird found in Australia. But the idea is these little bowerbirds, they're these little birds that make ground nests called bowers. The male will build the nest and the male will collect different things. And each male has a different favorite color, you know, some like orange, some like blue, some like red, some like green, and they'll collect different flowers and rocks and sticks, sometimes trash, and they'll build these really elaborate displays out front of their bower. And then the females will parade in front of the bowers and choose their mate based on whose bower they think is the prettiest. So that is an example of intersexual selection. And then again, natural selection cannot fashion perfect organisms. Natural selection is not goal-driven. The evolution of organisms is constrained. Selection can only act on existing variations. New advantageous alleles do not arise on demand, right? Mutations and things that cause these differences are random. And selection can only act on existing variations. It can't create new ones. It's an editing process, not a creative process. Evolution is limited by historical constraints. Evolution co-opts existing structures and adapts them to new situations. So, you know, there's something that has already existed in a population, and now we find that it might be useful for something we never even thought it could be useful for. But the environment's changed, so now whatever that is, it's useful. Adaptations are often compromises. The same structure often performs many functions, and then chance, natural selection, and the environment interact. Environments often change very unpredictably. All right, so you should now be able to explain how mutations in sexual reproduction produce genetic variation, describe the 5 conditions required for the Hardy-Weinberg equilibrium or for the Hardy-Weinberg principle, explain why the Hardy-Weinberg principle is significant to understanding the evolution of natural populations, define genetic drift in gene flow, understand the bottleneck effect and the founder effect and its effects on microevolution, explain how genetic variation is maintained in populations, and explain why natural selection cannot produce perfection. Why is there no such thing as the perfect organism? So that is the end of this lecture. I will see you for the next one.